In this video, we will discuss about the GlycoRNAs. That this researchers has found out that GlycoRNAs are also localized in the exosomes. So how they come up to these conclusions, we will discuss the results of their experiments in this video. So first, what they did is they separate out the fractions of the cells into the cytosols and nuclear fractions, and then check the presence of GlycoRNAs, and they are able to see that GlycoRNAs are mainly present in the cytosols but not in the nuclear regions. And they did the Western blot to confirm their fractionation quality, that FDH is the marker for the cytosols and the histone is the marker for the nuclear. Thereafter, once they find out that the GlycoRNAs are present in the cytosols, so they further fraction these cytosols into the soluble cytosols and the membrane associated cytosols. And in this case, they are able to see that most of these GlycoRNAs are found in the membrane associated cytosols, and some of them are found on the soluble cytosol regions. And similarly, that they did in the panel A, they also checked the fractionation qualities using the Western blot, and they are able to see that the SEC63 is the marker for the membrane associated cytosols. The GAPDH is the marker for soluble cytosols. So their fractionation qualities are also good. Thereafter, doing their literature research, they are able to see that they are able to hypothesize, or they come to a point that some of the GlycoRNAs might also be present in the extracellular vesicles. So they more precisely in the exosomal RNAs, in the exosomes. So they purify these exosomes and check the RNA contents. And since from the previous results they are able to see only GlycoRNAs only in the AC4 GalNAc treated cells, so they use only those treated conditions and check the cellular RNAs and exosomal RNAs. So in this case, they do not, in the ETBR, they do not see significant results for the exosomal RNAs, but when they check it for this after SPECT treatment through the biotin, they are able to see the exosomal RNAs. They are able to see GlycoRNAs in the exosomal purified regions that we can see here. So most of the RNAs that are present in the exosomes are GlycoRNAs, but they are not the regular RNAs. And this is the merged image, and here also they use the Western blot to confirm their fractionation efficiencies, that exosomes markers is CD9 and GAPDH marker is for the cell extract. Thereafter, to further validate the exosome purities or exosomes fractionation efficiency, they use this super resolution imaging of exosomes, extracellular vesicles using Strom microscopy, and they are able to see the average diameter of around 52 nanometers, which is also in line with the exosomes diameter. Then thereafter, what they did is they do the nanopore sequencing of the GlycoRNAs from the exosomes, and the results are shown here in the panel E, where we can see that the diameter of the circles indicates the length of the RNAs, and thereafter they also did the sequencing for the cellular RNAs also, and they can find out stark similarities between helacellular RNAs and exosomal RNAs. Around 52 RNAs are found common in both of these conditions. So this together indicates that most of these GlycoRNAs are mainly transported or transferred to these exosomes, and this is what they have come up from the results that are presented in this figure. I hope you understood how they did their experiment and what are their thinking in order to proceed in these directions. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. And these results are not in opposite to the previous GlycoRNA results that are presented in the that are presented or that are published earlier, because this results also shows that GlycoRNAs are mainly also found in this membrane associated cytosols fraction. But uh, this this experiments also give out a new finding that GlycoRNAs are also exported into the exosomes. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.